Okay, so thank you everyone for coming. Happy Thanksgiving week. So yeah, I'm a new assistant professor, relatively new assistant professor in chemical and biomolecular engineering, and my group is interested in problems at the interface of cell biology and sort of mathematical and computational approaches rooted in the physical sciences. So what I want to do today is tell you about sort of the way we think about some problems, tell you about a couple specific problems that I've worked on and we're currently working on. And so I'll say if there's anything jargony in the talk that's not clear, hopefully I'll introduce everything. But if you have questions, please feel free to stop me and ask. So I'm going to talk about signal transduction, which I'll define more precisely in a bit, at the cell membrane. Okay, and so the motivating feature today for the talk is thinking about T cells. So I'm sure many of you have heard through other researchers at NIMBioS about T cells. So T cells are cells of our adaptive immune system. They're the cells, one of the cells that are responsible for identifying pathogens and responding to pathogens that we perhaps have never seen before. And so what you're seeing in this image, there are 4 cells colored in red. There's another cell colored in blue. So the 4 cells in red are T cells, and these are interacting and identifying an infected cell in this case. Okay? And so the perspective we take and the question we want to ask is, how is it that these cells, these T cells, are able to use networks of biochemical, let's just say biochemical networks, okay? So whatever that means. They can be reaction networks, they can be biochemical protein-protein interactions. How do they translate these networks of interactions into reliable cell responses that allow a T cell to sort of reliably identify an infected cell? Because we're interested in the cellular level and how these individual molecular level events translate into cellular response. Okay, and so the key is looking at networks of biomolecular interactions. And so let's zoom in just a little bit now so we're all on the same page and think about how is it that cells interact with each other or with their environment. And the way they do that, if we take a sort of zoomed in view of a cartoon of the cell membrane is a lipid bilayer, so you have lots of different types of lipids. And associated with that lipid bilayer are many types of proteins, an order of thousands of types of proteins. Okay? These can be transmembrane proteins that span the entire membrane. These can allow the passage of material through the membrane. They can be molecules that are simply associated with the membrane. And the class of molecules we'll be interested in today are transmembrane receptors. So these are proteins that can bind to molecules outside of the cell and transmit information about that binding inside of the cell. Okay? And this is how T cells and other cell types identify things about their environment and then make decisions about them. And so how does a T cell translate sort of a receptor binding event into a response? Well, the picture is the following. Okay? And so let me walk you through this picture because I'll show it a few more times. What I'm showing here in dark brown in this image is the cell membrane. This thing right here called the TCR is the T cell receptor. This is a huge protein receptor. So this blue part is outside of the cell. This brown part is inside of the cell. And so what happens is you have this thing labeled peptide MHC right here. Just think about this as a chemical in the environment that's actually presented on this infected cell. So this is what contains signatures of pathogens. Okay, so basically through interacting with these peptide MHCs, a T cell has to decide, is this cell infected or is it not infected? Okay, and to do that, it translates this engagement here into a series and cascade of biochemical reactions within the cell. Okay, this is known as signal transduction. And so one of the central questions we're interested in is how is it that signals are propagated in space and time within a cell? Okay? And so what you roughly see here, this is kind of just a cartoon of what biologists think is going on. So you see these different circles and ovals. These are different types of proteins. You see lines connecting the proteins. Those represent interactions between the proteins. So for example, you see here, let's pick one, this thing called RAS-GRP has an arrow pointing to RAS. That means this activates RAS, and that means something specific in the context of this biochemical reaction network. You can also have inhibition or negative interactions. And so through these complex interactions, you can get things like feedback loops, complicated interactions that give rise to interesting dynamical and regulatory behavior. And so we're interested in understanding how these behave in both space and time. Great. So again, just to kind of recapitulate that whole picture, here's a bigger version. So we have, in general, an environmental stimulus. This is kind of the unified view of signal transduction that leads to receptor engagement. That means this receptor binds to this ligand in the environment. That leads to a signal that's propagated by means of various proteins within the cell, and that eventually changes the cellular response. Okay? And that's ultimately what we're interested in. How do cells make decisions, and then what do they do about it? Okay, and so again, just to sort of redefine this, signal transduction is the propagation of molecular signals by means of biochemical reaction networks. Okay, and by reactions, this can be things like proteins binding to each other. Proteins can chemically modify each other, so you can add things like phosphate groups or other chemical species from one protein to another. Okay, so that's the picture, and sort of the why mathematical modeling, why computational modeling? Well, these networks operate in a noisy environment. So we have thermal noise, we have fluctuations in numbers, and they really are organized across multiple time and length scales, ranging from receptor engagement, which is on the time scale of seconds, to cellular response, which can be on the time scale of minutes to hours, and ranging from the protein size, which is on order of a nanometer, up to cell sizes, which is an order of tens of microns. So that kind of gives you the context of what we're thinking about here. Yeah, of course. Yeah, please.
So each one of those little components in there,
Yeah.
does that correspond to really a well-known compound, or is there a whole collection of compounds in some of those circles that it's really...
So it depends is the answer. So for this T-cell signaling network, this is well enough studied and well enough characterized that each of these corresponds to a single protein for the most... Yeah, we'll say that's true.
So for example, the neck one,
Sure. This...
two outputs,
That's...
that means that there's two reaction products going on?
Yeah, so what this means here, so so this means, for example, this might bind to multiple, so these proteins are large structures that have binding domains, so it can bind to multiple types of proteins, and then it can chemically modify multiple proteins. Yeah, so it's not uncommon for a protein to have multiple targets. So it's not necessarily a case that this is a direct product, like a decomposition of this. It's that this protein interacts with this protein in a way that leads to some sort of activity. But for this network, every circle is a well-defined feature. The things that are less clear is when you see these interactions. Those are the things that are harder to determine, sort of from an experimental standpoint.
So we should or should not believe the output?
So you should not. That's the rule of thumb. Start from that perspective, right? That's the safe thing to do. Okay. Okay. So let me just give you a quick overview, since we're all neighbors and we're all part of UT here, and I just work a few buildings over. So my group, roughly, we organize ourselves around using theory and computation, so theory that's rooted sort of in theory from the physical sciences, to understand problems in cell biology and immunology. We think about a few different types of problems. This problem of signal transduction, which I just mentioned. We also think about the cytoskeleton, which is the network of biopolymers inside of the cell that gives it sort of its structural integrity and the ability to move. And we also think about biophysics of membranes, and I'll touch on that at the end of the talk today. And so the general question is how do these networks of interacting components give rise to cellular function? So that organizes how we think about things. Okay, I want to return to this T-cell problem one more time. So this is exactly what I just showed you, and tell you why T-cells are so incredible. In a sense, all cells are. But so these T-cells, which are identifying this infected cell, are specific, they're sensitive, and they're fast. And let me tell you what I mean by that. So each of these T cells expresses many copies of the same protein receptor, this TCR. So each T cell has on order of 30,000 of these expressed on its surface. Okay. So different T cells have different T cell receptors, meaning they have different specificity. So this is what allows different T cells to respond to different pathogens. Okay, so there's specificity. And the really amazing thing is that as few of these 30,000, as few as 1 to 10 need to be engaged with sort of a ligand that's derived from a pathogen in order to activate a T cell. When I say activate a T cell, that just means kick off the events that are required to get this T cell doing what it needs to do, get rid of the pathogen. Okay, so it does this on the time scale. It makes its initial decisions on the time scale on orders of seconds, although the entire process is much longer. It requires very few molecular signatures of the pathogen. And if you just change sort of a single point mutation in the pathogen signature, that will abrogate the ability of the T cell to identify. So it's very specific for a particular antigenic signature. Okay, so I want to tell you about two problems today. Time permitting, I think we'll get through these. So the first is I want to ask, how does the presence of the membrane influence properties of signal transduction networks? Okay, so we want to think about the spatial component of being near a membrane. And then the second problem I want to talk about is also related to membranes, and that's how do mechanical properties of membranes help to modulate the spatial organization of proteins at the surface. And I'll sort of introduce each of these separately, although they're connected in the broader view of how we're approaching these problems. Okay, so the first problem I want to mention is that of signal transduction. And something that may or may not be appreciated depending on your background is that biochemical reactions are inherently stochastic. Okay, so the traditional viewpoint for chemical reactions is you'll take a chemical reaction network, which writes down all these different chemical reactions, and you write an ordinary differential equation for the time evolution of each chemical species. These are coupled, usually nonlinear ordinary differential equations. Okay, this ignores fluctuations, so it ignores noise, and it ignores spatial component. I mean, one could write down a PDE model to get spatial components, but it turns out in a lot of biochemical systems, the presence of noise is important, as I'm sure all of you appreciate. And so I'll just tell you about the framework with which we think about this. And the reason stochastics are important, I mentioned as few as 1 to 10 receptors might be engaged. So you can already imagine you have small numbers, so any fluctuations will be large compared to the mean. And so we need to think about these as stochastic systems. And so what we'll do is we'll imagine that we can define the complete state of our system by this sort of state space vector, which we'll call gamma. So that represents all the information we need to identify the current state of the system. And in the language of sort of the physics and chemistry world that I come from, we say that we describe the dynamics by a chemical master equation. But I'll note this is just a continuous time Markov chain. Okay? And so the notation I'll use is that we have P of gamma t. This is the probability of being in state gamma at time t. Okay? And we'll write the time evolution of this probability as a sum of what look like gain and loss terms. Okay? So we have a transition rate matrix W, which tells us the rate that we transition from gamma prime to gamma times the probability of being in state gamma prime. This tells us the total rate at which probability flows into state gamma. Okay? And then we'll have the corresponding loss term, which tells us the rate at which sort of probability leaves the state gamma, going to all their possible states gamma prime. Okay? So this defines the dynamics of the system, although all the details, of course, there's no details here. All the details are embedded in W, right? So that tells you everything about the chemical reactions in the system. So I'm not going to talk about detail about the form of W. If anyone wants to talk about it, I'm happy to, or we can talk afterwards as well. But there are well-formulated sort of ways to translate chemical reactions into reaction rates that appear in this master equation or this Markov chain. Okay. So we're also interested in modeling spatial aspects. And so everything when I talk about space today, what we imagine doing is taking our space and discretizing it into a lattice. So we'll use a cubic lattice in 3 dimensions. It'll have arbitrary shape. We aren't really too worried. And the basic picture is the following. We view proteins as objects that can hop from lattice site to lattice site. When they're sufficiently close to each other, when they're in the same lattice site, then they can react. Okay? And according to whatever chemical reactions are in this signal transduction network. Okay, so the dynamics here, same equation. Okay? So this state space now is just the possible values that gamma can take on is just much larger. However, it's still a countable state space, so we have the same continuous time Markov chain dynamics. Why is it bigger? It's because now we need to track where the proteins are, as well as the state that they're in. Okay? That's correct, yeah. So for example...
So it's essentially a 3D cellular column.
Yeah, I think that's probably a language that's not the language that people usually use in this field, but that's right. Yeah, people would call this like an agent-based model sometimes, or... Yeah, so the language we'll use is that of a sort of a kinetic Monte Carlo. These are, I don't know, these are different fields, call them slightly different things. Yeah. Yeah.
When you say large, how large?
It depends on the lattice here, right? So, I mean, honestly, these are all finite state spaces that we're considering. So a typical lattice might have anywhere from... Yeah. Yeah, I would have. I think we're talking more 10 to the 10 to the 100. We will never try to enumerate this, by the way. All right, so we're just, I mean, that, so that in a sense it doesn't matter. I mean, I say that lightly, it does matter, but the way we're going to approach this is through stochastic simulations. It turns out for a number of simple problems, one can use analytical methods to look at these sorts of problems. So I did this by means of formal introduction, and what we'll actually discuss now is how we actually approach this, especially for this T-cell signal transduction network. Okay, and that's going to be through simulation methods that give rise to stochastic trajectories that are consistent with the dynamics. Okay, and so we use this kinetic Monte Carlo methods. This is common in statistical physics. In the biology world, this is often called the Gillespie algorithm. Okay, and this is event-driven. So this is the continuous time aspect, which we'll get to in a minute. And then each simulation will generate what we'll call a stochastic trajectory. All right, so this is a trajectory, one possible realization of the system that's consistent with the stochastic dynamics. Okay, so let me just walk you through how the algorithm works. It's pretty simple. All right, so given that we're in state gamma, we need to determine the rate of every possible chemical reaction. Okay, we'll denote the rate of each individual chemical reaction by gamma sub I. Once we have each of these individual rates, we know a total rate at which the entire system will transition. Okay, so once we have all these possible chemical reactions, we now have a total transition rate. Okay, so this will tell us now we'll have an exponentially distributed random variable with rate gamma total. The time associated with distribution will be the time to the next event. Okay, so the way one calculates this in a simulation is just you pick something that you can do on a computer easily, which is a uniformly distributed random variable between 0 and 1, and you can calculate an update time, which we'll call delta tau. Okay, so depending on the total rate, if total rate is large, of course, your delta tau is small, and so you're making very small increments in time steps. If gamma total is smaller, then you can make larger time steps. Okay, so you choose a time step in your simulation. So you're going to update the system over an interval delta tau, and then you need to choose what reaction occurred. Okay, and the way one does this is you pick reaction I with the probability is equal to the rate of that reaction over the total rate. Okay, and this gives you, this is just all based on the properties of exponentially distributed random variables. This is straightforward. Once you choose a reaction, then you update the state of your system. You get a new state, gamma nu, and you repeat this process. All right, you repeat it many, 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 many times, and then you have a stochastic simulation trajectory. Okay, so here's the idea. So we're interested in, I'm going to get back to the T-cell problem now, which is we're interested in what happens near the membrane. So what I'm going to talk about today is having a slab geometry. Okay, what I mean by that is we're going to have a system that's characteristically large in 2 dimensions, and will systematically vary that the length of that third dimension. So we're going to have this parameter we call L. We'll call this the confinement length. So basically, this is a length that tells us how much we're squeezing our system. Is it thick or is it thin? Okay, the thinner it gets, the more two-dimensional effectively it looks like, until we get to a single layer of lattice sites. Okay, so we're going to systematically vary the shape. We'll also systematically vary the mobility of molecules, so proteins diffuse more slowly at the membrane, and that's the reason for doing this. And this gives us a conceptual framework to think about the role of the membrane in signal transduction. Okay, so we're interested in stochastic effects. We're interested in the fact that the membrane is 2-dimensional, which might affect sort of diffusive properties of molecules compared to a 3-dimensional environment. We're interested in transport, diffusion, the fact that concentrations, when you confine things, get higher, and then correlations, spatiotemporal correlations between molecules. So if 2 things are close at some time, that means for local times they'll be close. Okay, so we're interested in characterizing those sorts of things as well. Okay, so here's what we're going to do. We're going to return to the same network. This is a zoomed-in view of that T-cell reaction network. And we're going to look at a very specific component of this. Okay, so we're going to look at this molecule called LCK. This is what's known as a kinase. A kinase is a protein that adds phosphate groups to other proteins. Okay, so this kinase modifies multiple sites on the cytoplasmic tail of this T-cell receptor. So when this T-cell receptor is bound to something in its environment, this kinase comes and adds phosphate groups here. Okay. Once those phosphate groups are added, then what happens is other molecules get recruited to the T-cell receptor. Those have their own enzymatic activity that then go and modify other proteins. So this is the very first step in the activation of a T-cell. Okay, so that's the story. And the idea is understanding this in its sort of membrane environment is important for actually understanding how T-cells work. Okay, and this is still an open question in biology. How is it that T-cells translate this engagement into a reliable response? Okay, so this is a system that can exhibit bistability. So this is, I'm sure, something that's familiar to all of you, so I'll just briefly go through it and why it's used in cells, or why cells use it. Okay, so bistability in signaling networks is often used in decision making. Okay, it's used, for example, in cell division. So once a cell decides to divide, it wants to keep dividing. It's used in cell death. Once a cell decides to die, it wants to make sure it dies. and in T-cell activation. Okay, and so sort of the classical picture in the energy well frameworks, this is more for the physics audiences, is you have two wells and your energy landscape is a function of an order parameter. These are deterministically stable, but one can see stochastic switching. Okay, and so the reason cells use these and the reason they're useful is an external perturbation could take you from one basin or one stable point to another, and then when you remove that perturbation, you stay in the new state. Okay. So let's look at a specific model of this phosphorylation. Okay, so we're going to take this picture of the cytoplasmic chain here, and we're going to translate that into a protein that has two sites. So this is just a cartoon of a protein now. Each of these sites can be chemically modified. Okay, we're going to view this kinase as sort of the light-colored Pac-Man and the phosphatase as a dark one. A phosphatase, I mentioned what a kinase was. A phosphatase goes the other way. It's a protein that removes phosphate groups. Okay, so here's the cartoon of what happens. We have this protein. It can bind a kinase, so one of these sites will bind it. It can then either unbind or it can chemically modify that site. Okay, so that's the method. And so if we write it out as a chemical reaction network, this is what I mean by a chemical reaction network. Okay, so basically the picture is the following. We have S0. S0 will represent this unphosphorylated or doubly open circle molecule. It binds to the kinase, E, this enzyme. It can either unbind or it can catalyze the production of S1. S1 is a singly phosphorylated protein molecule. S1 goes to the same type of reaction. S1 binds to a kinase. It can unbind or it can be catalytically modified so that it forms S2, and that's what we call our fully phosphorylated molecule. So the substrate molecule can be state S0, S1, or S2, and we'll count the number of each of these. Okay. We have our phosphatase reactions, which take us the other way from S2 to S1, S1 to S0, and then we have this sort of refractory period where the enzymes are inactive. So when they're E star or P star, they're locally inactive, and they have to be activated before they can rebind. Okay, when I say distributive mechanism, which I'll say again, this means that the enzyme has to unbind after each catalytic step. So there's some mechanisms known as precessive, where this enzyme can modify it to become S1 but stay bound to the protein. Okay, so that's our chemical reaction network. Is that clear? Hopefully.
Going back to your market share.
Yeah.
So basically, that means that there are like 17 reactions to keep track of.
That sounds correct. 1, 2, 3, 4, 5, 6, 7. Yeah. So we're actually. Yeah, that's. That's well, the size of. Well, the size of the state space would be dictated by the number of molecules in the system. Yes, that's right. But to actually calculate things, you need to know how many reactions there are, right? So that'll go into the calculation of the transition matrix. That's right. Yeah. This is a T cell's. This is inside of the T cell. Yes, this is. That's correct. So this is like what's happening just on the inside of the T cell membrane. Yeah. Okay, so why does this system exhibit bistability? Well, okay, so one can show rigorously that it in fact does exhibit bistability in some parameter regimes. Okay, this was discovered about a decade ago initially. Okay, and so what we'll do to characterize when bistability is present in the system, we'll characterize one of the stable steady states as inactive and one as active. So inactive is the state in which most substrate proteins, these S proteins, are unphosphorylated, which means that the number of unphosphorylated molecules will typically vastly exceed the number of fully phosphorylated ones. And so schematically, we'll show it like this. Most will be open circles, and we'll have maybe a few filled circles. The active state will just be the flipped state. Okay, and we've chosen the reaction rates here. Well, it doesn't matter. So we've chosen these. There's a symmetry between these states in the system based on the choice of reaction rates. So here it's the other way around. So most of them are fully phosphorylated or active, if you will. So they're able to recruit the other molecules to the membrane. Yeah, of course. Yeah.
So before you do kind of stochastic dynamics, it makes sense to look at what happens in the deterministic limit.
Yeah, that's right. Yeah.
So what happens in your system? It looks very simple, right? You can write like ODE.
Yeah, exactly. So
A little bit like two equilibrium, and that's basically it.
that's it. Yep. Yeah. So you can show the range of parameters here. I mean, it's relatively straightforward. It's a little cumbersome, but you have 1, 2, 3. You have on order of 10 chemical species. So you have 10 ODEs approximately. Yeah, and you look for steady states of those, show that you have multiple stable steady states. Yeah.
So there will be indeed multiple, more than two?
2. When I say multiple, I mean 2. Yeah. And this will have a maximum of 2. Okay, so sort of physically, the intuition behind this is the following. So the bistability arises because of a sequestration effect. So just for the sake of argument, imagine we're in the state where all of our proteins are unphosphorylated. So if this is the case, then we're going to bind largely these kinase enzymes. Okay, so that means we're going to have an excess of phosphatase in the environment. So the first sort of phosphorylation event that happens, let's say it's here, this first molecule becomes phosphorylated. The most likely next event that happens is not to bind one of these because there's very few, but to bind one of these. Okay. Binding one of the phosphatases will tend to take you back to that initial state, right? The phosphatase takes away what you just did. Okay. So there's certain regimes where this sequestration effect will drive the bistability. Okay. And so what happens when we actually simulate this? Okay, finally we get to a simulation result. So we have the following. So I'm going to show you trajectories. This is as a function of time on the horizontal axis, the number of fully phosphorylated molecules. I'm showing 2 stochastic trajectories. Each of these starts from the same initial conditions. And owing to sort of short time fluctuations, one sees 2 states that emerge. One is this active state, so most molecules are phosphorylated. The other one is the inactive state where there are very few molecules phosphorylated. Okay. And so what we'll do then to monitor for presence of bistability in the stochastic system is to look at long times, sort of when you've reached a steady state, presumably, or hopefully. I mean, we test for steady state. So you wait for sufficiently long times, and you monitor the distribution of molecules at this time point. Okay. And so what do we see? So the first thing we'll do is we're going to take the system. Remember, we had this confinement length. We're going to start with a thick system and systematically confine it. So going from thick to thin, we're doing that as we go from left to right. And if we then plot a histogram of the number of the number of active molecules or fully phosphorylated molecules at long times, we see what looks like a unimodal distribution for a sort of thick system. And as we confine it, we see the emergence of 2 distinct peaks. Okay, so this is associated with the underlying bistability in the system. So So each of these, I think it's 500 independent simulations. So basically what I've done is repeated. Here are 2 simulations. So this would be 2 data points that would be, so we would look at the very last time point, look at the number of molecules here and here. We do this 500 times, and then we'd plot the distribution on this histogram. So the frequency of observing 40 fully phosphorylated molecules would be 40 of our trajectories. Yeah, okay. Okay, so here's the basic picture. We confine the system, okay? As we confine the system, the concentration goes up. I should say we fix the number of molecules in the system. Okay, what this does, this increases the effective concentration. And the idea here is as you increase concentration, you promote binding between proteins because they're closer together. It's easy for them to interact. That enhances the sequestration effect that's necessary for bistability. Okay, so this is primarily a concentration-driven effect. So this next result I like. And here what we're going to do is we're going to keep the concentration fixed. So we're going to have 2 systems, one that's purely 2-dimensional, one that is slightly thicker. So we're going to shrink it by a factor of 2 in each of these big dimensions and increase it by a factor of 4 in that sort of thick dimension. Same volume, same number of molecules, different shape. Qualitatively, what I want you to look at is that the shape of this distribution is different for these 2 systems. Okay? So this is, all right, well, here's the first takeaway. So we know that bistability is not simply a function of concentration. Okay, so this can't be a concentration effect. And just sort of the takeaway here is molecules, on average, are farther apart in this purely 2-dimensional system than they are in this system right here. Okay, so the distribution of distances between molecules, in addition to the concentration, is an important effect here. Okay, and so if we put everything together, we now have our confinement length here. So as we go from right to left, we're making it thinner and thinner. As we go from bottom to top on this vertical axis, we're increasing the mobility of molecules so they diffuse faster and faster. And what I'm plotting here is whether or not it's bistable. So this upper regime is bistable, this lower regime is monostable, and the color code tells you how well separated the 2 peaks in the histograms are for this bistable regime. And the takeaways are that if you increase the mobility, you promote the bistability, and if you increase the confinement, you promote the bistability. Okay, so that's kind of. units are area down here. Here. Yeah, so a diffusion coefficient is always measured in these units. Sorry, yeah, so that's correct. So we're fixing the number of molecules. I'm not plotting concentrations, I'm plotting the confinement length. So the concentration will, well, yeah, so as L gets lower, the concentration goes up. This is the diffusion coefficient, so regardless of dimensionality, that stays the same units. Right? So square root of dt tells you roughly how far you go in a given time. Okay, just some examples. So you see the emergence of these more distinct peaks as you confine or as you increase diffusion. Okay. So why is this the case? So I just want to go through the effects here, and this is something we've thought a lot about. So I'll just kind of touch on the effects here. There's actually some really interesting mathematical problems here that perhaps one of you may be interested in thinking about or helping us think about at some point. So here's the idea. As you decrease the diffusion, okay, the time it takes for molecules to encounter each other goes up. And it's well known that the time that this takes can actually influence the kinetic parameters in a model. So the slower the diffusion, you basically decrease the effective parameters. Right? By decreasing the effective binding rates between molecules, you suppress the sequestration effect. So in a sense, qualitatively understand this. So decreasing the mobility decreases effective kinetic rates. So you get smaller fractions of bound enzyme substrate pairs, which we can see sequestration, okay, which then suppresses the bistability. So this is well understood for three-dimensional systems in which you have sort of very simple reactions. But to actually get quantitative estimates of the effective kinetic parameters for arbitrary geometries or more complicated reaction networks is a pretty active and interesting area with, I think, some fairly deep analysis techniques that are being used to study. So this is an interesting area that we're interested in thinking about more. Okay, so you decrease effective binding rates. Furthermore, it's known that rebinding, and I'll say what I mean by that in just a minute, can influence emergent behavior in three-dimensional systems. And so the idea here is that, let's look at this picture. So this is our substrate protein and our kinase right after this first phosphorylation event, okay? So the idea here, once we start to think about space, these are spatially proximal, all right? So once that reaction happens, they're still close together in space. So what that means is if we look at this reaction, S0 just went to S1, these are still close. And what that means is that if this enzyme becomes active fast enough, this will promote this reaction relative to what you'd expect by a well-mixed sort of non-spatial simulation. And so this is what we'll call a spatiotemporal correlation, the fact that when this happens, they're spatially close. And so this is kind of a nice exercise in random walks. And so, well, I'll talk about it in just a minute. So rebinding will change this distributive mechanism to a precessive one where you can go from S0 directly to S2. It's known that those types of reactions, one can show that those don't support bistability. And so by promoting this direct transition from S0 to S2, you decrease the presence of bistability in the system. Okay. And so this is a sort of a nice problem where you can think about the effects of diffusion and the effects of dimensionality and how that affects rebinding between these molecules. And so this becomes an exercise in thinking about random walks in 2 or 3 dimensions. Okay. So I think I'll just quickly show a couple of results. And so we've done some analytical calculations to think about this, and we're interested in the probability of rebinding a catalysis. Okay. So we're interested in the fact that if this just happened, what's the probability that this happens with the same 2 molecules? Okay. And so we've developed some methods that use ideas from first passage processes, Markov chains, for 3 dimensions and 2 dimensions. And so one can just quantify then as a function of concentration of molecules and as a function of diffusion coefficient, the probability that these 2 molecules come back. And so clearly it's suppressed in 3 dimensions compared to 2 dimensions. Okay. So here the idea is, well, in 2 dimensions, diffusive paths are much more likely to bring you back together than in 3 dimensions. And so anyways, this is another interesting area that we're actively pursuing. So the ways to think about how do these spatiotemporal events happen and how they affect sort of emergent behavior of signaling networks. Okay, so the ultimate takeaway is the following. As we go from this sort of 3D-ish, this thicker system that's cytoplasmic-like, high diffusion, large system, effectively 3 dimensions. And we confine ourselves to a 2-dimensional membrane environment, slower mobility, increased concentration. We see the following effects. So increased concentration enhances binding. Our decreased mobility lowers effective kinetic rates. And then additionally, we have enhanced rebinding owing to both mobility and dimensionality. So we get this sort of 3 different effects working. And these same physical features influence all membrane proximal signaling networks. So we've done some work with other networks as well. Okay, so this was actually work I started as a postdoc, just to give a quick interlude here. With Arup Chakraborty at MIT, we worked with a couple of groups at Berkeley and a couple of groups at UC San Francisco. And this, yeah, so these are the publications that contain some of this work here. Fun project, and still motivates a lot of problems that we're thinking about now in signal transduction. Okay, so I'm going to transition to the next part, which will be shorter, and talk about how we're thinking about ways that molecules are organized at membranes. Okay, and so the picture here is the following. We're going to go back to the same picture we had. So we have our T cell, we have our infected cell. And so when these 2 cells come into contact, there's actually a number of interesting events that happen. It's not just molecular binding. Okay, so this is now a different view of the same picture. So this is now the T cell on top. This is the T cell receptor binding to this peptide MHC that we talked about earlier. Okay, and so an interesting feature here is that the T cell has molecules of different lengths on its membrane. Okay, and so what happens is that if short molecules bind together, that prohibits longer molecules from entering that spatial region. Okay, so you have long molecules, short molecules, and so this can promote a spatial segregation of molecules of different natural lengths on the membrane. And so just to kind of show you what happens then is T cells are known at longer time scales. This is on order of minutes, so say 3 to 20 minutes. So the proteins on the surface of a T cell become organized. And so if we now look at this green thing as being the infected cell, the T cell on top, and now we look at this interface between the 2 and look at it face down, one sees that early on you see isolated clusters of molecules that eventually become spatially organized into this large, what's called immunological synapse. Okay, so the synapse is on order of 1 to 5 microns. This is big on cellular scales. It's big on the scale of a molecule. And so you see this large-scale organization, and there's open questions as to what's driving this. And the speculation is it's a combination of different effects, including things like molecular binding, membrane mechanics. The cytoskeleton most likely plays a role here, that you have active polymer reorganization of these things. Okay. And so what I'll tell you about today is some early work that we've been doing, thinking about some recent experiments, thinking about the biophysics of membranes and how they can help to spatially organize proteins at cell-cell interfaces. Okay. And this, of course, is all thinking about the bigger picture of how do cells interact and how do cells make decisions. So ultimately, we like to integrate this sort of framework that we're thinking about here with the previous signal transduction framework. And this is something that I think, well, it's actively being pursued. Okay. So here's the experiments. So this is kind of busy, but let me walk you through it. So this is based on a fairly recent paper by the group of Steve Boxer at Stanford. Okay, and so here's the experiment they do. They put down what's called a supported lipid bilayer. This is just a bilayer like a cell membrane. They then bring a big vesicle, so a big bilayer sphere. Okay, this is like a pretend cell, if you will. And they do something kind of cool. What they do is they can stick these DNA molecules on the surfaces of these bilayers. They design the DNA so that each of these is single-stranded here and here, so that when they get close to each other, they're complementary to each other, and they bind. Okay, so this gives you a synthetic analogy, if you will, to the receptor-receptor binding in cells. So what they find then is that these zip up together. Eventually, this vesicle ruptures, and you can have one tethered membrane, or one membrane that's tethered to the other. Okay, and then they can use different microscopy techniques to look at the distribution of molecules. Okay, so this, the takeaway here is this provides an experimentally controllable system that in a sense mimics things like the T cell synapse formation and other sort of cell-cell interactions. And so we're viewing this as a nice place to think about modeling these membrane systems. So what I want to do today is kind of tell you about the approach we're taking. Let me show you a few more results first. So here's the idea. The really cool thing they do now is they'll take DNA of different lengths. So they'll take a 24mer, so 24 base pairs that are complementary, 72 base pairs that are complementary. Clearly, this one's about 3 times as long as that one. And what they can do then is watch how these molecules segregate in space as a function of time, okay, in a well-controlled laboratory setting. And so what you see here, these are their experimental results. Okay, so the green represents the short molecule. The red represents the longer molecule. Okay, and so you see this is at initial time here. You see now at short times, you start to see the emergence of 2 domains, one that's green, one that's red. Where it's yellow, that's green plus red, so there's overlap there, right? And so as time goes on, you see a stronger emergence of domains. So eventually you see green domains in a sea of red. Okay, so this is short molecules in a sea of long molecules that eventually, over longer times, do what we call coarsen. And by coarsen, we mean start to coalesce. Okay. So this is the experiment. This is motivating what we're thinking about now. Okay. So I want to tell you about how we're thinking about this. There's a number of open problems here, and I think this is an interesting way of looking at these problems. It's been used fairly widely in a number of contexts, but I think this will be a fun way to look at cell-cell interactions. Okay, so here is our, if you will, our physical theoretical model. So we're going to have a continuum description of the mechanics of a membrane. Okay, so I'm showing you down here. This is that supported membrane that doesn't really move around much. And then we're going to have this tethered membrane on top. Okay, we'll describe the shape of that membrane by a function z. x1 and x2 will parameterize the position on the lower membrane. Z will tell you the height of the membrane on top above particular position x1, x2. So this is basically a height profile of the membrane. Okay. So in continuum mechanics and statistical mechanics, a typical way to think about this is thinking about what's called a free energy. Okay, so a free energy is basically an energy that tells you about how the system evolves in time. So as well, things tend to minimize their free energy. That's the idea. Okay, and so here we're going to have this free energy as a functional of the shape of the membrane, okay? And the way we write this is it's just an integral over x1, x2. We have a term that depends on surface tension. This term penalizes the creation of area, okay? So the smaller the surface area, the better. This second term represents a bending rigidity. This penalizes bending of membranes. So membranes like to minimize area, and they like to minimize bending. Okay, so those are kind of 2 rules of thumbs for membranes. So for a totally free vesicle or a soap film or something like that, this is the free energy that one would attempt to minimize by choosing a shape that's consistent with this free energy. Okay. Great. So now, how do we look at the idea of coupling these 2 membranes? What we're going to do is we're going to take different molecule types. So again, we have our supported membrane and our tethered membrane. We have molecule B1 and A1. These are the short DNA molecules. They can bind to form complex C1. We then have long molecules B2 and A2 that can bind to form complex C2. Okay, so we write these as chemical reactions like this. Their binding rates depend on the local separation between the 2. And if we want to look at the concentrations of B1, B2, A1, and A2, we write what are PDE versions of reaction diffusion equations. Okay, so here we're not going to be thinking about stochastic effects yet, or well, by yet I mean not in this talk, like maybe next year. Okay, so we have the concentration of molecule AI. It depends on a diffusive term. Okay, minus the reaction between AI and BI. So this is a chemical reaction term. This is known as mass action kinetics. And then plus an unbinding. So if this complex C1 unbinds, it will form an B1 and A1. Okay, these are reaction diffusion equations. Okay, how do we account for the interactions between these complex molecules? Well, we modify this effective free energy. Okay, so what we do is we have the same 2 surface tension, bending rigidity terms. We now add 2 additional terms that penalize. Here's the story. So we're going to have an energy penalty term multiplied by the concentration at a particular location times the square of the distance between the 2 membranes minus the natural length of complex 1. Okay, so what this tells you is that if complex 1, this short complex, is at a region of the membrane that is not compatible with its natural length, we'll incur an energy penalty here. Okay, this is just sort of the simplest energy penalty that one can write in this free energy. And this is consistent with a lot of sort of molecular features of real molecules. Okay? And so that's the idea. We now have a modified free energy that couples these two membranes. Okay? So how do we handle this time evolution of these complexes? There's sort of I guess one would call it a standard methodology in statistical physics. It's known as time-dependent Ginzburg-Landau theory. This is sort of part of a field of physics known as field theory. And let's just look at the time evolution of the height profile. This tells you how the height of the membrane varies as a function of time. And we simply write the following. So we write that it is a functional derivative of f with respect to z. This basically tells you have a gradient descent, that the membrane changes its shape to try to minimize the free energy. Okay, this is a way that's consistent with many dynamics. Okay, so that's the picture. And we have a similar picture for the complexes. So let me actually tell you what we do now. Okay, so these problems are amenable to analytical theory in some cases, but they're also relatively easy to study with simple numerical methods like finite difference. And so I'm just going to show you a result. I'm going to show you the membrane height profile. This is x1, x2. The color tells you the distance between the two membranes. Okay? We have equal concentrations of A1, B1, A2, B2 at initial times. And then we basically will solve this this will show you 400 seconds at sort of physical parameters of what happens. Okay? So we have some initial small fluctuations in membrane shape, and we see the following. Oops. Yeah, there we go. Okay, so this is what's known as a coarsening process. Okay, so this now we're seeing a coarsening of the two systems. We see an emergence of a red domain, which is a large distance between the two membranes, and a blue domain, which is a close distance between the two membranes. And over time, we see these sort of coarsening, or if you will, becoming more distinguished for blue versus red. If we look at the concentration of molecules, it will exactly mirror this. Okay? So now we're looking at the concentration of the short complex. So we expect this to be high, or red, where the membranes are close, or blue over here. And in fact, we see an almost direct correspondence. Okay? So we have a means, through computational methods now, to solve these equations using finite difference, standard techniques, and we can see the emergence of dynamics that are consistent with some features of the experiments, time scales, length scales, etc. Okay. One can play with things like what happens if you change the relative concentrations. Okay, so now we have, let me do this, get this lined up. Okay, so now what you're going to see is we have more short molecules than we have long molecules, and we're going to see the emergence of these islands of the longer molecules in the sea of these shorter molecules. Okay, so this now gives us a way to sort of explore parameter space using these computational methods. Okay. So one thing that was quite surprising to us was that in the process of doing this, well, let me actually just give you the quick overview. So we see a fast patterning to small domains, okay? And then we see a slower coarsening. So that is, you see the initial sort of segregation followed by a much slower, larger domain formation. And the relative concentrations of the short and long molecules influence sort of the qualitative features of what we see. Okay, so one thing that was really surprising to us is that these simulations suggested that the bending rigidity of the membrane didn't play a major role in the dynamics of this system. Okay? And so we turn to analytical methods to look at this, and I'll just kind of give you the overview. So again, this is the same free energy here, okay, that we wrote down earlier. And so now what we're actually interested in is how can we treat this in some analytical method? And so there's ways to functionally integrate this. And so in statistical physics, one can write down what's known as an effective free energy. Let me tell you what this means. So we're going to functionally integrate the exponential of this functional, okay? And this will basically tell us something about an effective free energy. Physically, what happens? This tells us, imagine we don't want to keep track of the shape of the membrane anymore. This gives us a way to average over all possible membrane shapes in a way that's consistent with the energetics of the system. Okay? This effective free energy now will only depend on concentrations. It will no longer depend on the shape of the membrane. Okay? So there are ways to do this exactly, and so when gamma and kappa are 0, one can solve this exactly. And what one sees is that this effective free energy term has some constant terms which aren't particularly important. And you see here now a coupling between the concentration fields. And so we see a C1 times C2 under this integral here. And this tells us that our concentration fields are coupled even in the absence of these elastic moduli. Okay? And so this gives us a driving force for molecular segregation even when the mechanics of the membrane aren't playing a role in the system. So this, I think, is something that hasn't been appreciated in the literature yet. So this is something that we're currently thinking about, the role that these may or may, the role that gamma and kappa may or may not be playing in cell interactions. Okay, so quick overview. The interplay between molecular binding and mechanics is quite common. Happens anytime two cells come together. These field theoretic models capture key features of the experimental phenomena. Okay, so we're exploring and trying to push these further. And then also these calculations we do suggest that even in the absence of mechanical properties of the membrane, just having a coupling between the molecules is enough to drive spatial segregation. Okay, and so just again, this is the overview of my group. We're all friends here, so it's great. I'm looking forward to talking to more of you over the years, and I want to thank my group. This is Sina, Paul, Rob, Benny, Eva, and Aaron. Benny and Eva are two talented undergrads. The rest are grad students. And then for the first part, these are the T-cell signal transduction collaborators. So thank you very much for your time and your questions. Thanks.